Hi guys, this is Lab 15 review, female reproductive system for Histo. Again, Histo is generally our best taught subject, so mostly making this for ease of review during exam 2. But we'll get some physio out of this, I think, and let's get to it. So we are looking at the ovary first, then we're going to be looking at the uterine tube, the fallopian tube, and then the uterus, the cervix, and then we end with the then we'll look at the vagina, and then we end with looking at the mammillary glands of the breasts. So, starting with the ovary, we know, of course, that the ovary is responsible for the production of oocytes, right, the eggs. And in particular, we have, this is a cross-section of our ovary. You can see that we have a whole bunch of what we call follicles. And these follicles are going to eventually become our oocyte, the egg, right? And so we're going to trace it along different stages of these follicles, and we'll see them go from our primordial follicle that females are born with, and it's stuck in prophase of meiosis 1 as a primary oocyte in the center of this follicle, and then around it, it's got squamous follicular cells, right? So it's a primordial follicle. Under the influence of FSH, then we get these follicular cells are going to become granulosa cells right there. You still have the egg on the inside as an oocyte, primary oocyte. Our cuboidal granulosa cells, which had been previously follicular cells, but follicular stimulating hormone as a pulsile release from the anterior pituitary caused this change to occur. We said it's about 10 primordial follicles are going to undergo this change every month, and we'll end up with one overall follicle makes it. All right, so after we've become a primary follicle as a result of FSH, then we have continued growth of granulosa cells, meaning they're going to divide, and so you go from a unilaminar single layer of granulosa cells to a multilaminar, which you can see here. And now you have a primary follicle that's multilaminar. Still, the oocyte in the center is a primary oocyte. I don't know why they don't show the nucleus here, but it would be there. Then you have the zona pellucida has been made by the oocyte. Then you have the addition of the granulosa cells are going to produce a liquid, and that's going to accumulate inside of the follicle as the antrum. And so once you have the appearance of an antrum, you now have a secondary follicle, but still a primary oocyte in the center. All right, and we have our zona pellucida. All right, then you get to just more production of that follicular liquid from our granulosa. So we get a bigger and bigger antrum, still a secondary follicle, but this is going to end up as a Graafian follicle, which is secondary. It's going to complete meiosis 2 when it ruptures, or it's going to complete meiosis 2, it will rupture, and then eventually become the corpus luteum during ovulation. All right, but all of this stuff occurs in day 1 through day 14, and we'll talk more about that in just a minute. All right, but we're not that far yet. We're looking at the ovary itself, and inside of the ovary we have this nice cross section with our follicles. All right, so on this ovary, more superficial to the follicles, we have this layer of dense irregular connective tissue called the tunica albugina, and on top of that you're going to have a mesothelium. All right, deep to the tunica albugina we do have our follicles. Again, if it's primordial, we're going to be seeing simple squamous on the outside. All right, so this appears to be simple squamous. It's really hard to tell because it's not zoomed in very well, but simple squamous primordial follicles. And then what is all this other stuff around here? This is just stroma. Why do we care about stroma? Well, our stroma are going to become our theca interna and theca externa cells. All right, so this is more of what we just said. We have our primordial follicles, right? So these are the ones you're born with. You notice that they're surrounded by simple squamous, and these are follicular cells. Here's the oocyte, and then under the influence of, and you can see these here, right? The oocyte in the center. We have nice squamous follicular cells on the outside. You have, so then if they are not squamous, then it's not a primordial follicle. It's past that. So anyways, what takes you from primordial to primary follicle? Well, we already said it is the first step of our ovarian cycle. And so you may recall that the pituitary releases FSH and LH in a pulsile fashion, all right? And so the first part of our pulse for females is the release of follicular stimulating hormone from the anterior pituitary into the blood. That's going to be taken up by our follicular cells, which are going to become granulosa cells as a result. So step one, FSH is released by the anterior pituitary as this pulsile release. And then follicular cells become granulosa cells, and you're like, okay, well, why is that important? Well, we're going to have some baseline level of androgens in our system, and we have our granulosa cells take in follicular stimulating hormone, and they take in an androgen in the form of testosterone, and they produce estrogen. All right, and so now you have granulosa cells producing estrogen, and the estrogen is going to help with follicular growth, and so then you're going to have the oocyte develop, more granulocytes or granulosa cells are going to go from single like this to more like this, stratified. All right, so we have now our primary follicles as a result of FSH, and we have the creation of a zona pellucida by the oocyte, and so we're past primordial follicles as a result of the first step of the ovarian cycle, the FSH pulse from the anterior pituitary, and that first step has caused us to go from primordial to primary follicular. All right, and then now we have the ability to secrete estrogen, and so we're going to continue along this path, and you can already see there's more and more granulosa cells, and you're going to see estrogen start to skyrocket. All right, but we still need our theca interna cells, so we're going to talk about those in just a second. All right, so here's more of what we were just saying. We've gone from primordial follicle to primary follicle, and you can see that we are no longer simple squamous. So our follicular cells, right, these guys are now nice granulosa cells, which are estrogen producing under the influence of FSH. All right, and then just a reminder that inside of these guys we have our oocyte, and all of our oocytes are stuck in meiosis 1 prophase until it becomes a Graafian follicle, where it will do meiosis 2, or it will finish meiosis 1, and within 12 hours it will have ovulated. All right, so primary follicle, we saw here's unilaminar, here's multilaminar, and you can start to see a zona pellucida around the oocyte. All right, so Graafian follicle, which is the next step here, it's a secondary oocyte, is what a Graafian follicle is. It's massive, completes meiosis 1 about 12 hours before ovulation. Okay, so after you've finished being a primordial follicle, we said that the granulosa cells are doing a couple of things, but one of them is they are producing a liquid folliculi, and so that accumulates inside of the follicle. And so once you start to see this sort of giant space, we'd call this the antrum, you now have a secondary follicle, all right? And so, during this and with the production of estrogen, you also have the stroma that has been outside of the follicle is starting to differentiate, and so you're going to get stroma is going to become theca interna and theca externa, all right? And so it's kind of jumping ahead, but now we have our secondary follicle. We still have our zona pellucida around our primary oocyte. We have our granulosa cells here. These are mural granulosa cells around the edge. This section right here is the cumulus oophoris, which is a mound of granulosa cells, and then a part of it right here around the oocyte itself is the corona radiata. And then again, these are mural cells on the outside. Then we have a basement membrane right here, and outside of that is former stroma. So we have theca interna cells right here, and then theca externa cells right there, and then more regular undifferentiated stroma around it. Our theca interna cells are going to be stimulated by luteinizing hormone to produce testosterone. All right, and so we have really a two-cell system here in the ovary. Our luteinizing hormone that's in the blood is going to come bind to a receptor on our theca interna cells, which you can see here, and here are our granulosa cells. And binding to this luteinizing hormone receptor, it's going to mediate the uptake of cholesterol, which will then be converted through a series of chemical reaction steps by the theca interna steps into testosterone. So testosterone, aka an androgen, is going to be released into the blood. So there's some blood vessels here, and then also to the granulosa cells that are adjacent to the theca interna cells. And then the theca interna cells, or then the granulosa cells, will use aromatase to convert the testosterone into estradiol. They'll do that under the influence of FSH from the blood. All right. You'll notice here that we also have a receptor for LH on the granulosa cells, and this is important because, as we know, we have a pulse of FSH at the beginning of the ovarian cycle, and then we're going to have a pulse of luteinizing hormone at ovulation. And so that's going to cause the granulosa cells to undergo a change, and it's going to cause ovulation. And so the fact that there's a luteinizing hormone receptor on the granulosa cell is actually quite important for ovulation and the changes that are associated with that. All right, so outside of the theca interna cells, which are right here, we have our theca externa cells, and those are contractile overall. All right, so what happens? We said that we started with 10 primordial, and we end up with 1 graphian that eventually ovulates. So what happens to the other ones? Well, they become atretic follicles. Atretic follicles are the ones that basically they die. It gets infiltrated, hard to see here, but we have like a bunch of macrophages, lots of connective tissue. This is a super thick basement membrane right here. And so this is an atretic follicle. It's a secondary follicle that didn't make it. All right, so we have gotten a little bit further. We are now on the secondary follicles. We're just about ready to ovulate. All right, so again, what was our ovarian cycle so far? We had follicular stimulating hormone as a pulsile release from the, right, happens like once every month, pulsile release from the anterior pituitary. And that's going to cause our follicular cells, right, these squamous cells, to become granulosa cells. All right, those guys are going to start releasing estrogen as a result of aromatase working on testosterone. Then we're going to have the stroma cells are going to differentiate into theca interna and theca externa. And the combination of the theca interna and the granulosa cells are going to allow for a great production of estrogen. So our theca interna cells, under the influence of luteinizing hormone, are going to take in cholesterol, produce an androgen in the form of testosterone. That will then be taken up by our granulosa cells under the influence of follicular stimulating hormone, and that will produce estrogen. All right, and so we're seeing that as our follicle grows with more granulosa cells, we're getting an increase in estrogen, right? So this is stage 2. All right, and then we know estrogen is going to stimulate follicular growth, right? So just more of this, our follicle's getting bigger. And then it's also going to stimulate endometrium growth, which we're going to talk about a little bit later, but we're not quite there yet. All right, and then finally, it's going to have a negative feedback on the release of FSH and LH overall. And so, you can start to see, negative feedback. Negative feedback is going to lower FSH and LH after we have a pulse, right? All right, so we got our giant secondary follicle. Now we have a pulse of LH, again, pulsatile. It just happens once a month from the pituitary glands. That pulsile release is going to do 2 things. We have that LH is going to come, and remember we said there was an LH receptor on the granulosa cells. And so the LH receptor on the granulosa cells is going to be, it's going to decrease estrogen production, all right? Whereas follicular stimulating increase the granulosa cells production of estrogen, right? And so Dr. Braun gave us a good way to remember this. He just said that the granulosa cells were originally follicular cells, right? And so follicular stimulating hormone is going to stimulate granulosa cells to produce estrogen because a granulosa cell used to be a follicular cell, right? It's still inside of the follicle, but it's been with the follicle the entire time, right? So luteinizing hormone is going to have a negative feedback, decrease estrogen, right? That's what we're seeing here. Luteinizing hormone saying, hey, granulosa cells, chill. All right, the next thing it's going to do, it's going to get that secondary oocyte, aka at this point we'd call it the Graafian oocyte, to finish meiosis 1 and ovulation occurs. All right, so we have ovulation has now occurred, and you have, we'll talk about what happens with the corpus luteum in just a second, but you also have in addition to this binding the receptor, the theca interna and the granulosa cells are now going to become cells of the corpus luteum. And so theca interna cells become theca lutein cells, and granulosa cells are now granulosa lutein cells. All right, and so theca interna cells previously were all about testosterone production, right? That's what we see up here. Granulosa cells were all about estrogen production. That's what we see up here. But now that they've changed into corpus luteum cells, they are now all about progesterone, pro-pregnancy, making progesterone instead, right? And so progesterone is going to grow the endometrium overall, as we'll see in just a couple of minutes. All right, so importantly though, we have the follicle has ovulated, which means the Graafian cells, a lot of the Graafian cells stay behind, a lot of the theca interna cells stay behind. The corona radiata granulosa cells, go with the oocyte, which is now a secondary oocyte. But you have the remaining behind are most of the granulosa cells, and then all of the theca lutein cells, right? And so that's these guys. And it's going to start off as a corpus hemorrhagia. It's going to be full of blood. It becomes a corpus luteum, and then that corpus luteum, as we said, is going to be, now it's going to be made up of theca lutein and granulosa lutein, and those are going to produce progesterone, which is going to have an effect on the endometrium. But this guy right here had the luteinizing hormone causes change, and so luteinizing hormone this will be maintained, maintained by luteinizing hormone, but we have a problem here, right? We have a pulsatile release of luteinizing hormone, so this guy's going to go down. So this guy, corpus luteum, is going to die unless we have new luteinizing hormone. All right, and so this is problematic because we're not going to get any more luteinizing hormone, but we could get HCG if our female becomes pregnant. And so if there's no luteinizing hormone, which there won't be, and no pregnancy, no HCG, then you have your corpus luteum is going to die. But if you do have pregnancy, you have the placenta is going to make HCG. HCG is identical in function to LH, close enough in structure that it's the same thing. And so HCG will maintain the corpus luteum, and the corpus luteum produces progesterone, pro-pregnancy, aka pro-endometrium, which will mean it will maintain the endometrium of the uterus. Okay, all right, so we can see here that the progesterone is rising as a result of the corpus luteum, and then estrogen is also rising again. Why would that be the case? Well, we have our granulosa cells are mostly producing progesterone, right? You can see it's higher, but they are still producing some estrogen. All right, so corpus luteum is the fate of our follicle, right? It's the granulosa cells and the theca interna cells that are left behind. So luteinizing hormone stimulates the ruptured Graafian follicle to become corpus luteum after ovulation. Luteal hormones prepare the uterus for implantation during day 15 to day 28. All right, HCG is going to prevent its degradation past day 28. And we said granulosa cells become the granulosa lutein cells, and theca interna become theca interna cells, or theca lutein cells. All right, both are producing progesterone. All right, so here's just a picture of the corpus luteum, and the idea was our theca interna cells are going to come inwards. They're going to bring blood vessels with them. We just have a lot of granulosa cells here as well, mainly progesterone. Estrogen is going to dominate the follicular phase, and progesterone is going to dominate when the corpus luteum. All right, so follicular phase. Now this is the luteal phase where progesterone dominates. All right, so not too bad. Then, as we said, we have the corpus albicans. And so the corpus albicans is what happens if the corpus luteum, we said it's not going to receive any more LH, which is needed to maintain it. So this guy is going to die if there's no HCG to replace that LH. And so the corpus albicans is what's going to happen to the corpus luteum. So no pregnancy, no HCG, the corpus luteum degrades, progesterone and estrogen levels decline, and if these two decline, then you're going to get menses, and the endometrium will slough off, you'll have a period. Okay, important to know, this is our ovary, this whole thing, this is a corpus luteum, isn't it just massive? Crazy. All right, and then here's some scar tissue that is our corpus albicans. All right, corpus luteum, notice that it is, solid. This is scar tissue overall. And then remember, a atretic follicle is dead follicle, but it generally has some sort of lumen, so that's how you can tell it apart. All right, there's the occasional macrophage inside of here as well. And then the atretic follicle is much smaller. All right, so now we're past ovulation. And so we are ending up the oocyte, the secondary oocyte is going to end up in the fallopian tube, and where it will hopefully meet a sperm and then become a zygote, and then that zygote is going to implant upon the endometrium. So starting with the fallopian tube, notice how it is open to the abdominal cavity, but we have our first part, which is the infundibulum with the flimbrae. We have our ampulla, then we have the isthmus overall, and we're going to see it's a highly folded lumen. All right, so the uterine tube, right, this whole thing, uterine tube, also called the oviduct or the fallopian tube, is again where we said fertilization occurs, but not where implantation occurs. We want that to occur in the uterus, not the fallopian tube, right? If it occurred in the fallopian tube, that would be the ectopic pregnancy, which is quite dangerous. All right, so what's going on with the histology of the fallopian tube? Well, we have this very highly branched mucosa, and we have two cell types. So here we can see that we have a little bit of a fuzzy surface. So we have columnar. Our first type is ciliated columnar cell, which is to move the zygote along. And then we have these nice little peg cells, which is like the name of, which produces nutrients for sperm. All right, but they don't have cilia on them. And then notice that it does have a large lamina propria overall. This does look a lot like the seminal vesicles that's producing fructose for our sperm on the male side of things. But in comparison to the seminal vesicle, we have a lot of lamina propria. All right, so let me just take a look at that real quickly, see if we can find that. So let's see, seminal vesicle, accessory glands. Oh, that's not a good sign. All right, let's see. Oh. All right, seminal vesicle. All right, notice that the lamina propria is quite small, basically no connective tissue, whereas in the case of the female oviduct or fallopian tube, you have a lot of connective tissue. All right, so moving on, and hopefully our zygote is moving on as well into the uterus. The uterus has been undergoing some changes, which we'll talk about, but the basic idea is that there are 3 layers to the uterus. And again, so we started in the ovary. We had our secondary oocyte come here. Hopefully it met a little sperm. If it did, it's going to move down here into the uterus where it will plant on the endometrium, which is right here in this particular view. The endometrium is located right here. There are 3 layers to the wall of the uterus. So the endometrium, which is where the zygote would implant itself. And then we have the myometrium below it. It's a very thick muscle layer. And then below that, we have the perimetrium, which is a thin serosa. All right, so we'll start with the endometrium. We have a simple columnar layer at the very top. Don't have a good view of it, but you can see this little diagram here. Maybe that's better. The endometrium is going to have a nice functional layer, which gets built up during the proliferative and secretory phases. And then we have the basal layer of the endometrium, so functional layer, basal layer. Deep to that, we have our muscular layer, which we call the myometrium. And we'll talk more about that in just a minute. We do have uterine glands in this region, which are going to change throughout the endometrial cycle. And they are simple tubular, but basically we're going to have the functional layer right here is going to change, and this will get sloughed off if there's no pregnancy overall. All right, so functional layer right here, basal layer. We'll talk more about this, but the menstrual cycle, basically we have during period, the functional layer, right, this layer of the endometrium gets sloughed off during menses, right? So that's between 0 and 5 days. Then you have the start of production of estrogen by our granulosa cells, right? As we get more and more granulosa cells, production of estrogen, that's going to tell the functional layer of the endometrium to grow. Then you get to day 14 and you have ovulation. And so you're going to have a dip in estrogen, right, as a result of the LH acting on the granulosa cells. And then they're going to start producing progesterone instead. And so progesterone is going to take us from basically, a nice functional layer with straight glands to these, very big sort of spiral glands overall that are going to be ready for the implantation of the zygote. All right, but progesterone, estrogen. All right, so during the early proliferative phase of basically, this is the proliferative phase, and so we're around here. This is where the functional layer of the endometrium had been sloughed off. We basically just have basal layer here. We have some nice straight arteries, and we're going to start growing these spiral arteries, which we'll see will spiral up. And so we have not much of a functional layer yet. We're just right here. It's starting to be built up. You can see that there are some glands. It's not super obvious. This is really the function or the basal layer that we're seeing that's starting to grow. We have a simple columnar epithelium overall, and our goal is to rebuild the functional layer. All right, so we're going this way. Okay, then we are here in sort of late proliferative phase. We're more this way. You can see we had like barely any glands right there. Now we're starting to get more and more of these straight glands. And so here we have the functional layer. Here we have the basal layer. And I don't know if you can make this out, but we actually have, there's a difference in color here, right? And so this lighter color is our basal layer. Down here is our functional layer. And then deep to all of that is our myometrium. All right, and so this is still proliferative phase, right? This whole proliferative phase, aka the estrogen phase or follicular phase, and we see that the straight glands have been developing, and they're lined by simple columnar cells. We still have simple columnar at the top, and they're maturing with more estrogen secretion. All right, so functional layer is getting thicker, lots of straight glands. We can see all of these here. We don't have to be able to tell between the late and early phase, right? But the glands are much more pronounced than the early phase. And then there was just a quick note that Dr. Braun wanted us to know. So recalling that oocytes are only viable for 24 hours. So during ovulation, the fallopian tube will be dominated by the ciliated columnar cells to move the egg along. And then during the secretory phase, the PEG cells, and let's go back, our PEG cells are releasing nutrients. So during the secretory phase, they're going to release a lot of nutrients. Those are in the fallopian tubes, but the same logic occurs here, right? When we're in the secretory phase, close to implantation, we want to produce a lot of nutrients for the impending implantation of the gamete. All right, or zygote. All right, moving on. So it's the same thing, right? We're proliferative phase. We're not really excreting anything from our straight tubules, but when we get to the secretory phase, now we're going to start excreting stuff. All right, so you can notice a big change here, right? Straight tubules, now we're under the influence of progesterone, right? This is the progesterone uterus. You see we have all these nice squiggle lines, and so our functional layer is here. We have our basal layer here of the endometrium, still have our myometrium down here, but we are going to be releasing a lot of glycogen in particular. So over the next 14 days after ovulation, the functional layer of the endometrium is becoming bigger. You get these coiled glands with plenty of secretory activity. It's a mucus filled with a lot of glycogen. All right, so here we have our functional layer, our basal layer. Again, you can tell some difference in the color. And our straight glands have become coiled glands, which are secreting nutrients now. All right, so again, we are in the secretory phase now. All right, so what happens if we do not have implantation and the generation of a placenta, and our placenta makes HCG? So if you have no pregnancy, that's no HCG, which means no equivalent to LH. So no LH equivalent. No LH equivalent, right? Our LH levels have already dropped because it's a pulsatile thing, but we need an LH to maintain the corpus luteum. So if our corpus luteum is no bueno, that means no progesterone. We said this is the progesterone uterus. So what if we don't have any progesterone? Well, then we are here and we are going to have degeneration of the corpus luteum. All right, so menses phase, we have, torn vessels, blood in the stroma is what we're supposed to see here. But here we have our basal layer. This is our functional layer. It just looks red and gnarly and it's not good. So open vessels, glands torn, hemorrhage, right? Glands, functional layer has sloughed off. All right, so moving on. Now we have the cervix. So it's kind of abrupt, but all right, so endometrium of the uterus, and then the end of the uterus ends in the cervix, and then we have the vagina. So the cervix inserts into the vagina, as we just said. It has cervical exocrine glands, which are secreting mucus onto the surface, and this exocrine function. During the follicular phase, it is sperm friendly, so it's going to produce a thin and alkaline mucus. During the secretory luteal phase, it's not going to be sperm friendly because, right, we had a small window of 24 hours at day 14 where that sperm better have done its job, and if we're past that, right, we're implanting now, so sperm is no bueno. All right, so anyways, epithelium is simple columnar, and we have two parts of the cervix. We have the endocervix with these nice mucus glands, and then we have the ectocervix, which is adjacent to the vaginal canal. And so here we have the cervical canal, here we have the endocervix, here we have the exocervix overall. All right, and it's bending around, and then it's going to come and in the vagina right there. Okay. All right, so more on the cervix. We have, as we said, a simple columnar epithelium, which we could see here. The exocervix, which is right here, is going to have stratified squamous, non-keratinized. And so the explanation I heard from this was, why would you want stratified squamous? Well, the vagina is the receptacle for the penis, right? And so there's friction. And so the cervix, being adjacent to the vagina, is going to need a friction surface. And so you have stratified squamous, non-keratinized. And so you can see a transition from, columnar to stratified squamous, non-keratinized. And so this would be your start of your ectocervix, right there. All right, then we're in the vagina, and as we said, receptacle for the penis. So stratified squamous, non-keratinized. Interesting thing here, though, is notice how pale staining this is, and sort of vacuated, I think is the word we would use. There's no glands in the lamina propria, but why is it so vacuated looking? For the vagina, estrogen promotes the storage of glycogen, and so basically you're going to, as epithelial cells are exfoliated, glycogen is released and metabolized by bacteria producing lactic acid. This lowers the pH in the lumen and inhibits growth of pathogens, and so this is interestingly a defensive mechanism. So allowing for some useful bacteria to kill off other pathogens. All right, but deep to the lamina propria, we do have a muscularis externa. So we don't have it really easy to see here, but we do have an inner circular and then outer longitudinal layer. All right, and then a loose connective tissue adventitia. All right, so we're almost done. This has been a long one. Mammillary glands. So now we're dealing with the breasts. We have these lobules of compound tubular alveoli glands, which are going to produce milk. We have basically terminal ductules right here. And these are going to be simple cuboidal epithelium. And then those all merge to become our intralobular ducts, which are going to be double cuboidal epithelium. And then those will come together to be our lactiferous duct, which is going to be stratified cuboidal epithelium. So basically we're going from one to more and more layers of cuboidal epithelium. All right, so lobules, terminal ductules, intralobular ducts, lactiferous duct. All right, and so we're going to have some changes during pregnancy overall, but we do have some different units. So we said that we had the terminal ductules right here, and these are going to be in contact with the alveoli, and those come together and form our lobules. And so we said, these are meant to be kind of a single layer of cuboidal overall. Then these guys are multiple layers of cuboidal lobules, intralobular ducts, and then lactiferous ducts are going to be, several layers of stratified cuboidal overall. All right, there is going to be myoepithelial cells around the ducts that are going to contract with oxytocin, and in our physio lecture we said that oxytocin can be released as a result of suckling. All right, so simple cuboidal epithelium, and then myoepithelial cells on the outside. We are going to have differences based on non-pregnant breast, pregnancy, and then lactation. And so in a non-pregnant woman, the gland has the non-pregnant woman, the gland has an inactive duct system. During pregnancy, the glands proliferate, and you're going to have the formation of alveoli, producing a concentrated milk colostrum. And then during lactation, the alveoli are fully differentiated and can produce a lot of milk. All right, so prolactin is going to stimulate milk production, and oxytocin, which we know can do contractions in the uterus, will also stimulate milk release. Okay, so we're going to have a difference in what the histology slides look like. All right, so starting with the inactive mammillary gland, you can see we have some lobules, doesn't look like very much, lots of adipose, lots of connective tissue overall. There's some lobules, but they're small and separated by interlobular connective tissue. So the lobules are mostly ducts at this stage, right? It's mostly ducts. And we have a lot of dense, irregular connective tissue overall. And then, as we said, a lot of adipose tissue. All right, when you have a pregnancy, notice a difference already between this and this. Right? We have, as we said, the alveoli are starting to grow, but we do have a lot more ducts, so more glandular cells, parenchyma, functional cells. There are lots more lobules, and they are larger overall and closer together, less connective tissue. Inside of the lobule itself, right, you see connective tissue here, but there's much less connective tissue. It's hard to tell, duct versus alveoli, but we were told that you can sometimes see a little bit of milk. So right here, this appears to be some milk. This would be an alveoli. And then again, cuboidal cells for ducts. So simple cuboidal is probably a duct. All right, and then we still have our myoepithelial cells. They respond to the oxytocin. All right, here's lactating. Notice how the adipose tissue is not nearly as prevalent, and you have a whole lot of gland overall, right? You zoom in, you can also see a lot of milk, right? So this would be lactating. Here's milk. And then a reminder that milk lipids are released by apocrine. Some of the cell goes with it, and then proteins are merocrine secretion, no loss of cell membrane. And then a friend again, prolactin stimulates the production of milk. All right, so we've reached the review section. This is a lot. So primordial follicles, We said have squamous cells on the outside. Unilaminar, this would be unilaminar primary follicle. This looks like it might, this I'd say still unilaminar. This is clearly not unilaminar. I don't see any antrums. This is a primary, secondary. All right, so multilaminar, secondary follicle. So here we have an antrum. This is secondary. And you can see a little bit of zona pellucida there, which is nice, great. Theca interna cells, you can see are past the basement membrane, remember, because theca interna cells were actually part of the stroma. And here's theca externa cells, here's just stroma cells. This is an atretic follicle, so cool. Uterine tube, we said is highly folded, and it's going to have ciliated columnar cells, and then those PEG cells for nutrient production. And then here's a electron microscopy of primordial follicle. We have our oocyte, and well, this whole thing's the oocyte. This appears to be the nucleus, and a basal lamina, and stroma. It's a little bit hard to tell. Single layer flattened follicular cells. So presumably these are flattened follicular cells. Okay, so because this is a primordial follicle. All right, primary follicle. Oh, nice. Okay, so here's our oocyte. Here's our zona pellucida right here. These appear to be our granulosa cells. Here's our basement membrane, and then theca interna right there. All right, lactating mammillary glands, lipid dropules present, okay, and myoepithelial process that appears to be right here. Interesting. Okay, well, that's it. That was super long, but good job, guys, and we'll see you next time. Keep at it. Keep on keeping on. All right, bye, guys.